Good morning. Thank you, Maury, for the introduction. Mesoblast is a publicly traded company, so please note the forward-looking statement. Mesoblast was founded in 2004 and became public at that time on the Australian Exchange. We have maintained a strong financial position and retain about 50% of our investment base from institutional investors. Our company corporate strategy is to leverage proprietary cell-based and complementary biological technologies to develop products for unmet medical needs. We bring multiple products to market. Our aim is to bring multiple products to market within a parallel time frame and to underpin our future financial growth through investing in manufacturing operations and to enhance the likelihood of commercial success through strategic partnerships. We operate through a risk-managed business model. We're working with multiple products in parallel time frames. The products specifically target major medical conditions where proprietary technologies offer scientific and clinical advantages. We maintain a strong cash position, which enables simultaneous development. The commercial success is enhanced via strategic partnerships and growth through profitable manufacturing operations. And these offer the potential to deliver significant and sustainable revenues. The Mesoblast proprietary technologies are described on this slide. They include the Mesenchymal Precursor Cell technology platform for bone marrow and adipose tissue-derived products. We've also recently added culture-expanded Mesenchymal Stem Cell products, and I'll talk more about that later. It also includes Dental Pulp Stem Cells and expanded Hematopoietic Stem Cells, where the mesenchymal cell is the feeder layer for hematopoietic stem cell expansion. Additionally, we are developing certain biotherapeutics based on protein factors that are derived from proprietary cellular platforms. Our strategic partnerships are our partner with Teva Pharmaceuticals, primarily for the development of cardiovascular and neurological indications. We also have a strategic alliance with Lonza to ensure commercial scale-up for manufacturing for supply, product delineation, cost of goods reduction, and expansion of clinical manufacturing as needed. We've also recently added a collaboration with JCR Pharmaceuticals to develop MSC products in Japan for the indication of graft versus host disease in adult and pediatric recipients. As I mentioned last week, we acquired Osiris cultured MSC platform, and there are many strategic benefits related to this acquisition. They include firming up Mesoblast's position as a leader in regenerative medicine, accelerating our time to first product launch, broadening our market opportunities with new phase 3 programs, facilitating a leverage build-out of infrastructure, skills, and expertise that are needed for commercializing Mesoblast MPC products. This also expands our reach to Japan through the collaboration with JCR Pharmaceuticals and the acquisition of intellectual property, which is highly complementary to Mesoblast's already existing patent estate. Mesoblast has broadened the intellectual property estate through this acquisition. The acquired Osiris patent portfolio is highly complementary and additive to Mesoblast's existing patent estate, which covers composition of matter and uses in the US, Japan, Europe, China, and other jurisdictions. And so in addition to the existing 26 patent families, Mesoblast has acquired an additional 35 patent families from Osiris relating to the culture-expanded MSC technology. And specifically, those acquired patents comprise granted patents in the US and abroad, as well as patents extending through 2025 and applications out to 2031. The clinical benefits of acquiring the MSC's assets are many. We're broadening Mesoblast phase 3 ready products in two new indications. Those are Crohn's disease and acute graft versus host disease. Now, in Crohn's disease, we have acquired the ongoing 330-patient phase 3 trial in patients who have Crohn's disease and have failed other biological agents. And acute graft versus host disease, we've acquired the promising phase 3 results in adults with GVHD complicated by gut and liver complications. We've also acquired an expanded access program in pediatric steroid refractory acute graft versus host disease. And our plan is to obtain U.S. FDA approval for Prochymal, which is the only stem cell therapeutic designated as both an orphan and fast-track product. Prochymal has already received conditional approval in Canada and New Zealand for the treatment of children with acute graft versus host disease, and it's available in the United States, as I mentioned, under the Expanded Access Program for acute GVHD. And ownership of Osiris' extensive long-term clinical data from over 1,500 patients treated with cultured MSCs includes both safety and efficacy data, as well as extensive data on repeat dosing of MSCs in these indications. Mesoblast clinical development programs focus on products that use the MPC or MSC platform technologies and continue to operate within 4 major areas of development, as shown on this slide. They are as follows: systemic diseases of inflammation and immunity, orthopedic diseases of the spine, cardiovascular indications, and oncology conditions related to bone marrow transplantation. The combination of the MPC and MSC technologies places us with 6 late-stage clinical trial programs across the 4 core therapeutic areas where we expect in 2014 to be in phase 3 across multiple areas, as shown here. These include the Crohn's disease program, spinal fusion, degenerative diseases of the disc, heart failure, acute graft versus host disease, and a bone marrow transplantation with expanded hematopoietic progenitor cells. The immune inflammatory focus includes Crohn's disease, as I mentioned. This is a recently acquired program through the Osiris platform. Crohn's disease is an inflammatory condition of the gastrointestinal tract, which affects over 700,000 people in the U.S. with over 20,000 new cases annually. And there are approximately 60,000 patients who are intolerant, unresponsive, or refractory to existing therapy. Prochymal has shown benefit in early trials in Crohn's disease, and it's currently being evaluated in a phase 3 330 patient program of patients with moderate to severe treatment refractory disease. And following an interim analysis earlier in this program, the best performing Prochymal dose was selected to complete the study, and this was based on assessment of pre-planned assessment of the primary endpoint being disease remission. The other immune inflammatory programs that are in progress for the MPC technology are type 2 diabetes and related conditions, as well as rheumatoid arthritis. This year we completed enrollment of a 60-patient phase 2 safety study evaluating a single dose of intravenous MPCs in patients with early type 2 diabetes that is not adequately controlled with oral hypoglycemic agents. The patients were evaluated over 12 weeks for effectiveness of the single dose, for changes in glucose control, inflammatory responses including CRP. And data analysis for this study is about to commence, and results will be announced before the end of this year. We've also begun enrollment under Australian approval for a phase 2 study looking at diabetic nephropathy. Our rheumatoid arthritis program is enrolling. This is a U.S. program which is evaluating a single dose of IV therapy of MPCs for biologic refractory RA. We have 2 active programs in spinal diseases, and both involve treatment with MPCs with carriers. These are locally delivered products. The spinal fusion product has completed a phase 2 trial in lumbar fusion, and this study, the results have previously been disclosed, demonstrated equivalence of the MPC treatment for local fusion at 12 months compared to an autogenous autograft. And this was measured by radiographic fusion, pain reduction, and improvement in function. And so data from this study are currently being packaged and evaluated for discussions with the agency as an end of phase 2 discussion to move forward into phase 3 trial. So if successful, MPCs would be utilized for fusion and would eliminate the need for autograft and associated risk of pain, infection, and blood loss site at the harvest site. The degenerative disc program, the cells are injected locally with a carrier to regenerate the intervertebral disc. And we've completed enrollment in a 100-patient study in this indication, a phase 2 program. And we've recently looked at 6-month results from the first 50 patients in that indication. And the analysis of this showed that the single low-dose injection of MPCs resulted in significantly greater reductions in low back pain and improvement in function, as well as overall treatment success when compared with controls. And treatment success was a combined radiographic and functional endpoint. And full results from the 100 patients in the study followed up to 12 months will be available shortly and will undergo internal evaluation and then regulatory discussion. The cardiovascular program is one that I think most people are fairly familiar with. We conducted and completed a phase 2 trial of MPCs for congestive heart failure. This involved a single endomyocardial delivery of the MPC cells utilizing a catheter. And this study completed enrollment protocol, and patients have now finished a 3-year follow-up. And the complete data set has showed and continued to confirm the duration of effect in this indication, where the highest MPC dose, patients still have not experienced any hospitalizations for decompensated heart failure, nor have there been any cardiac-related deaths in this high-dose treatment group over the 3-year follow-up period. And this is an initial observation that we saw as early as 6 months following the treatment, and now see the duration of that effect, which is very distinguished from the number of events we've seen in controls, and the durations now out to 3 years for that effect. And so we have continued to work very closely with our commercial partner Teva to finalize the clinical plans for the phase 3 program with all the documentation for regulatory submission and review. So we continue to work with our partner to prepare to begin that phase 3 program. We're also conducting an ongoing phase 2 study looking at intracoronary delivery of MPCs for acute myocardial infarction patients for the prevention of heart failure in that patient group. And we have, through the Osiris data, acquired long-term safety follow-up data, which is ongoing for an acute MI study conducted in MSCs. And our programs in oncology are related to bone marrow transplantation indications, and they are acute graft versus host disease as well as expansion of cord blood. So allogeneic hematopoietic stem cell transplants are used for patients who have malignant diseases, immunodeficiencies, or genetic disorders that require transplantation for management of their diseases. For the most part, patients with hematologic malignancies are diseases where transplantation is required to prevent death from disease. And as you know, transplants are limited often by the availability of donors. And so there remain a number of patients who could benefit from bone marrow transplantations but who do not have an available donor. For many who do undergo allogeneic transplants, graft versus host disease remains a potentially life-threatening complication, and it arises in about 50% of all recipients overall. Particularly in patients with gut and liver complications of GVHD, the mortality, the morbidity is significant, and the mortality is high and can reach 85%. So in phase 3 trials, Prochymal was evaluated and demonstrated to significantly improve overall responses in adults with gut and liver GVHD, and those responses correlated with improved survival. And in phase 3 study, Prochymal also significantly improved overall responses and survival in children with a severe graft versus host disease. We're also in phase 3 program with MPCs for expanding cord bloods for transplantation. And so these are transplants that are utilizing 2 cords for patients who, for the most part, do not have another donor source for transplant. And for many patients, the dosing in cords are insufficient to provide acceptable outcome, and outcome in many cases is dose-related. And so even with 2 cord transplants, the results, particularly engraftment-related endpoints, are not equivalent to bone marrow or peripheral blood sources. So we've undertaken initially a phase 2 collaboration with MD Anderson, looking at using the MPCs as a feeder layer for cord blood expansion. And the results of that study were positive. They were published last year in the New England Journal of Medicine. And we've now embarked on a phase 3 program using MPC expanded cord blood to accelerate neutrophil and platelet recovery in patients who are undergoing transplantation for hematologic malignancies. So with that, I'll end with a thank you to the audience and thank you to the sponsors.